Hello, everyone, and welcome back to lecture number 2 of our Lipid Metabolism Series. Last time, we followed dietary fats from your plate into your enterocytes. But what about the fat that your body makes itself, even when you haven't eaten any fat? That's called de novo lipogenesis. Today, we are going to answer a deceptively simple question. How does a liver cell take two carbon fragments from carbohydrates and turn them into a 16 carbon fatty acid like palmitate? This process is absolutely essential for storing energy, building membranes, and even producing signaling molecules. But it's also a key player in metabolic disease like fatty liver, obesity, and insulin resistance. So, open your notebooks where we are going to build a fatty acid, carbon by carbon, enzyme by enzyme, and we will end with a clinical pearl that might save a patient's liver. De novo. De novo means from new. Unlike dietary fat that we absorb, this is the fat we synthesize, mostly in the liver, also in the lactating mammary glands, and a little in adipose tissue. What about the brain? No. What about the RBCs? No. They can make their own fatty acids. Let's talk about the de novo synthesis in much more detail, right? Imagine an assembly line. On this slide, I show you a cartoon of a liver cell. Acetyl CoA comes out of the mitochondria. We will see how. And then we need ATP as an energy and NADPH, the reducing power. Where does NADPH comes from? Mostly from the pentose phosphate pathway. That's why defects in that pathway affects fatty acid synthesis. The star of the show is the fatty acid synthase, FAS, a giant multifunctional enzyme complex in eukaryotes. We will walk through all 7 steps. So let's start with the production of cytosolic acetyl CoA. Acetyl CoA is made in the mitochondria from pyruvate via PDH complex and from the fatty acid oxidation, but it cannot cross the inner mitochondrial membrane. But how does it get to the cytosol? The answer is very simple, the citrate shuttle. Mitochondrial acetyl CoA plus oxaloacetate leads to citrate formation via citrate synthase. Citrate exits the cytosol, then ATP citrate lyase splits it back into acetyl CoA and oxaloacetate. And oxaloacetate goes back as malate or pyruvate, generating NADPH in the process. Draw the shuttle on the board. This is a very favorite question for exams and by point of view. So next is the carboxylation of acetyl CoA to malonyl CoA. This is a committed step of the fatty acid synthesis. The enzyme is acetyl CoA carboxylase, ACC. It adds a carbon dioxide to acetyl CoA using biotin as a carrier to form malonyl CoA. It requires ATP and biotin, the vitamin B7. The regulation point is ACC, that is acetyl CoA carboxylase, that is activated by citrate. that is high energy signal, and inhibited by palmitate CoA, the end product feedback, and AMPK, the low energy state. So acetyl CoA plus carbon dioxide leads to formation of malonyl CoA with ACC in the middle, right? So malonyl CoA is a 3-carbon molecule that will donate 2 carbon to the growing chain, losing the carbon dioxide later. Now let's discuss the multifunctional enzyme, the star of the show, that is fatty acid synthase. This is the masterpiece of biochemistry. In biochemistry, and especially in as if we discuss the bacteria, the FAS or the fatty acid synthase is a separate enzymes. In us, in humans, it's one giant polypeptide with 7 domains, like a Swiss Army knife. ACP is the swinging arm that carries intermediates. The step 1 to 3 is the loading step. The step 4 is the condensation. The key carbon bond forming steps, right? Catalyzed by ketoacyl synthase, and carbon dioxide from the malonyl is released. That carbon dioxide originally came from the carboxylation step, and its loss drive the reaction forward. The next reaction is the reduction, that is from the NADPH to NADP plus formation. And the next step is the dehydration. And the final step is the reduction again, NADPH to NADP plus formation. After 7 steps, a carbon chain is transferred back to cysteine and a cycle repeats, adding 2 more carbons each time using a new malonyl CoA, and after 7 cycles you get palmitate. This illustration shows the complete steps of this pathway. And at the end, you get the palmitate. Now, let's discuss the further elongation of fatty acid chains. As we discussed, palmitate is produced, and palmitate is not the end. In the endoplasmic reticulum, not cytosol, elongases add 2 carbon units from the malonyl CoA to make stearate, that is 18 carbon molecule, and arachidate, that is 20 carbon structure. This uses similar chemistry but different enzymes. Mitochondria can also elongate, but mainly endoplasmic reticulum does it. Very long chain fatty acids like that contains 22 to 24 carbons are important for brain sphingolipids. Now, let's discuss the desaturation of the fatty acid chains. To make unsaturated fats, like oleic acid, we need desaturase. These are endoplasmic reticulum membrane enzymes that introduce double bonds using oxygen, NADH, and cytochrome B5. Humans can desaturate at the delta 9 position between carbon number 9 and carbon number 10, but cannot introduce double bonds beyond the carbon number 9. So we cannot make omega 3 and omega 9 from scratch. That's why linoleic acid and linolenic acid are essential fatty acids for humans. Now let's see the regulation. Fatty acid synthesis is expensive. Lots of ATP and NADPH is used in this process, so it's tightly regulated. Short-term regulation includes ACC is allosterically activated by citrate, high acetyl CoA and ATP, and inhibited by palmitate CoA, that is the product feedback mechanism. Phosphorylation by AMPK, that is indication of low energy. The high AMP inactivates ACC. So long-term regulation gives the carbohydrate diet that leads to insulin production and that upregulates the ACC and FAS gene expression. Fasting or glucagon that leads to downregulation. Dietary fat suppresses de novo synthesis because you don't need to make what you already ate. Now let's discuss the clinical correlation, fatty liver and alcoholics. Now, for the clinical part, you do alcoholics. Why do alcoholics get fatty liver? Ethanol generates enormous amounts of NADH in the liver via alcohol dehydrogenase and aldehyde dehydrogenase. That high NADH to NAD plus ratio leads to that inhibits the TCA cycle because isocitrate dehydrogenase is NADH plus dependent. Secondly, that shunts the acetyl CoA towards the fatty acid synthesis. And thirdly, it inhibits the fatty acid oxidation because beta oxidation needs NAD plus. So what about the result? Massive de novo lipogenesis plus reduced fat burning leads to fatty liver. This is irreversible. This is reversible if the patient stops drinking. So, and that class is how your liver turns a ball of pasta into stored fat. For example, if you take glucose, that leads to acetyl CoA formation and ultimately the malonyl CoA, and that leads to the palmitate formation. So, what about the takeaways for your exams? ACC is a rate limiting committed step activated by citrate and inhibited by palmityl CoA and AMPK. Fatty acid synthase is a giant multifunctional enzyme that builds palmitate from acetyl CoA and malonyl CoA. NADPH comes from the pentose phosphate pathway and malic enzyme. Desaturases need oxygen. They are why we need essential fatty acid in our diet. Finally, alcohol induces fatty liver through NADH overload. So, next time in lecture number 3 of this series, we will talk about fatty acid oxidation, how we break down the fat we make. If this lecture made the carbon by carbon process finally click for you, hit the like button, share this video with your biochem study group and batchmates, subscribe to our channel, and click the bell icon so you never miss the upcoming lecture. I am Dr. Muzamil Liaqat Ali. Keep feeding your curiosity. It's the only thing that should be synthesized in excess. Until next time, thank you very much.